Drew, I will just mute yourself, like you, for the moment I'm talking, and then if I can. Yep. Okay. So welcome everyone. We are 30 participants so far. So welcome to all of you to the second webinar of our webinar series, the Research and Winemaking webinars. And so today we will talk about the management of sulfur dioxide in winery. And that's very important, especially right now, just because harvest will start pretty soon. Maybe it already started in some areas. And to avoid microbial spoilage, oxidation, different things in your grapes during the winemaking, you want to manage properly the sulfur dioxide. So if you didn't attend the previous webinar on winery cleaning and sanitizing, I'm Dr. Aude Watrelot. I'm an assistant professor of enology at Iowa State University in the Department of Food Science and Human Nutrition. And I'm co-hosting and co-organizing this webinar series with Mr. Drew Horton from the University of Minnesota, who is a research specialist of enology. Yep. So this webinar is being recorded, as I said. If you have any question during all the webinar, so you will have two presentations, and after we'll have a Q&A section, about 15 minutes. So please type any of your questions either in the Q&A box or in the chat box that is available. If you don't see the Q&A box, just click on the three dots at the bottom of your screens and you should see the Q&A box appearing. And so, as I said, we will answer your question at the end of this section. Also, we are always looking for feedback just to know how we can improve ourselves to provide you a lot of information. So please take a few minutes at the end of the webinar. You will receive an email with a link to a survey, a Qualtrics survey. So please just take time to complete this survey. So now, regarding the organization of the webinar, we'll start by the presentation of Dr. Gavin Sacks from Cornell University. So he will discuss about the proper and non-proper management of sulfur dioxide for about 20 minutes. And then we'll have Katie Cook from Scott Laboratories, who will discuss about the practical aspects of the good management of sulfur dioxide in a winery, also for 20 minutes, and then we'll have the question and answers. So that's my pleasure to introduce our first speaker, our first presenter, Dr. Gavin Sacks. So Dr. Sacks is a professor and associate chair of food science at Cornell University. He has a PhD in chemistry from Cornell University. His research interests are in developing and utilizing state-of-the-art approaches to analysis of trace level components associated with food and beverage quality, with a special focus on mass spectrometry-based tools. He teaches courses related to wine flavor chemistry and analysis, and has co-authored a textbook in wine chemistry. So Gavin, Professor Sacks, I will give you the presenter role, and then you will be able to share your presentation. Thank you.
Success.
Yep.
Wonderful. Good. So yeah, I'm gonna take your word for it, Professor Watrelot, that there's actually people out there, and I'm not just talking to you, Drew, and Katie. Unfortunately, I can't see who else is in the participants, but I expect that many of you are in the Iowa or Minnesota wine industry and make wine and have added SO2 at some point in your life. Let's see. Okay, so I was asked to speak on both the proper and practical use of sulfur dioxide in the winery. Some of the more proper and practical aspects are going to be covered by Katie Cook. I'm going to be talking about a little bit of that, but also some newer research, some improper and impractical recent work that we've done that may not be of immediate use to you, but hopefully will be in the future. Okay, so I'm going to do some quick review. Katie's going to talk a little bit more about the measurement side. I'm going to be focusing a little bit more about some of the theoretical aspects. So when we talk about SO2, we're talking about multiple forms of SO2. So what are these different forms? Free, bound, molecular. What do we do with these numbers? Because a measurement's only, from a practical standpoint, measurement's only as good as what you can eventually turn to something operational. What do you do with that number once you have it? And then I'll talk about some new work that we've been doing over the last few years, comparing what we refer to as apparently free SO2 versus truly free SO2. Okay, so free SO2 and wine. This may be review or maybe brand new. So we talk about sulfur dioxide and SO2. If you look in wine, if you were to add your potassium metabisulfite, for example, or bubble SO2 through solution, most of your SO2 is not actually SO2. It exists in this other form referred to as bisulfite, HSO3-, at least at wine pH. So we have two different forms of SO2 that contribute to free SO2. And those are referred to as bisulfite, HSO3-, and molecular SO2, which is just like the name sounds, it's S sulfur onto the two oxygens, SO2. And the ratio of those two is going to depend on pH, with molecular SO2 favored at lower pH values. So the molecular SO2, or SO2 just as SO2, is a pretty small proportion of free SO2. And the amount that we have in molecular SO2 is going to be generally less than 5%. It may be as little as 1% or smaller. And the higher the pH, the smaller the proportion of molecular SO2. And so in your textbooks or online, you might find calculators that will have, it's off the scale, but this is pH down here, that will have a relationship between pH and free SO2 and to achieve different molecular SO2s. So if you have a pH of 3.2, free SO2 at 50, you cross, and a molecular SO2 here is around 2 ppm. And these relationships can be found in a number of sources, or you can have online calculators to convert between the two. So why the emphasis? Why do we even bother concerning ourselves with these two different forms? One of the major reasons is that the two different species, different pools, molecular SO2, free SO2, have different functions in the wine. Molecular SO2 is the antimicrobial fraction. If you look at that structure, SO2, it's neutral, that is, it has no charge. It is able to diffuse inside of cells, including the cells of bacteria and yeast. And once inside the cell, it can serve as a broad spectrum antimicrobial. So the total amount of SO2, the free amount of SO2 in a wine is not a good predictor about SO2's antimicrobial activity. Rather, it's the amount of molecular SO2. So molecular SO2 is the antiseptic form, microbial form. Yeast, and especially Saccharomyces cerevisiae, are relatively SO2 tolerant, much more so than bacteria, like lactic acid bacteria or acetic acid bacteria. And depending on the presentation you go to, the textbook you're reading, the workshop you just finished up, you're going to see different recommendations for what target molecular SO2 you should be using. I'll let Katie go into this more. I presume that she's got some of this in her presentation. But generally, higher values, somewhere closer to 1 milligram per liter, are recommended for wines with more residual sugar because your main threat are yeast like Saccharomyces that are relatively SO2 tolerant, with lower values recommended for typical dry wines where your main threats are Brettanomyces yeast or lactic acid bacteria spoilage, or maybe even acetic acid bacteria spoilage. So constraints, why not just add as much as you want for molecular SO2 or have a really high level? One is that this is also the fraction that you smell, that you detect with your nose. If you ever put your nose over some potassium metabisulfite, you've probably noticed the struck match odor. There's a sensory threshold of around 200 milligrams per liter. Another constraint that people concern themselves with is you can inhibit malolactic fermentation. Okay, so the other fraction of SO2 beyond molecular SO2, or another fraction, the other role of SO2 is its role as an antioxidant. And I'm going to focus here on its role as an antioxidant in wine, although it also has a role with preventing oxidation in juice. Although some people will or will not add SO2 to juice, almost everyone adds SO2 to finished wines or to wines during storage. So free SO2 is what is reacting with the products of oxidation. So to be picky, oxygen does not directly react with oxidize every component of wine. Rather, it reacts with specific components in the wine, such as polyphenols, and it generates other species, hydrogen peroxide, quinones, and these can lead to reactions that in most wines are considered undesirable. There'll be a formation of oxidized aromas, browning, loss of desirable aromas. If you're making sherry, this isn't a problem, but if you're making most table wines, this would be considered to be a problem. So the role of SO2, and more specifically, the role of free SO2, is not to directly react with oxygen. Rather, free SO2 in the form of bisulfite, HSO3-, will scavenge, react with the products of wine oxidation. So you make hydrogen peroxide when you oxidize a wine. SO2 in the form of bisulfite can rapidly react with that hydrogen peroxide. You make these quinones, which are oxidized polyphenols. They can do terrible things to wine. They can result in a loss of, cause browning, cause a loss of good fruity flavor compounds. Bisulfite will react with quinones before they can react with other compounds in the wine. So put another way, SO2 is an antioxidant, but not because it reacts with oxygen directly. Rather, it scavenges the products of oxidation. Also, if you work through the chemistry, do a little math, you find out that for every milligram per liter of oxygen that gets into a wine, if it's all consumed, and you have the wine that starts off with SO2, then you expect to lose 4 milligrams per liter of total SO2. At bottling, typically you expect to have 2 to 3 milligrams per liter of oxygen. So to put this into perspective, at bottling, you've got a typical bottling line, you'll have enough oxygen in the bottle to consume maybe 5 to 10 milligrams per liter of SO2. Okay, so we've talked about molecular SO2 targets already. Typical free SO2 targets, again, pick your workshop, pick your textbook. Typically, free SO2 targets are somewhere around 30 milligrams per liter when you've got a wine that's being stored in some sort of package or I should say some sort of container, assuming you don't want it to go through fermentation. If you want malolactic fermentation, if you want a secondary fermentation, like for sparkling wine, these targets will be low. But if you want to prevent, if you're both trying to prevent fermentation and you're trying to avoid oxidized aromas, then this target of around 30 milligrams per liter is pretty typical. And once you get below around 10 milligrams per liter, wines begin to smell oxidized, even before you get to 0. So you want by the time that wine gets to the consumer, to make sure that your wine still has at least 10, probably a little bit more, of that free SO2 to make sure that it still smells acceptable. Okay, so we have 2 complementary targets for SO2. One is molecular SO2, which is the antimicrobial species. The second is free SO2, which is the antioxidant form. These are 2 complementary targets. You think about them, they're going to be dependent on each other, related by pH, but you can't only look at one and ignore the other. Generally, you're thinking about both. How much molecular do I have to prevent microbial spoilage? How much free SO2 do I have to prevent oxidative damage to the wine? Okay, so this also brings us to why it's, for most wines, not sufficient just to say, I've got a free target, why do I need to worry about a molecular target, or vice versa? Here in the Finger Lakes, we have dry white wines like dry Rieslings that are routinely pH 3.2 or less. 3.1, 3.0, there's some reason as low as 2.6. If you have a general strategy of, well, I always just make sure free SO2 is at 40 ppm or 40 milligrams per liter, you go to your lookup tables. Here we are. You're up at around 2 parts per million. That's the sensory threshold for molecular SO2. So fine, you've got plenty of free SO2. You've got so much molecular SO2 that people will be smelling burnt match and sneezing when they smell your wine. This is the kind of wine where you might want to dial down the free SO2 that you have in packaging and be careful with oxidation so that you can present an acceptable product. Okay, so there's a lot of interest in trying to limit the use of SO2, and you can limit the use of SO2. It is very hard to eliminate the use of SO2 in stored wines, whether it's stored in a tank, stored in a barrel, stored in a glass bottle, or some other package. There are options for molecular SO2 as an antimicrobial. Keep in mind, the brewing industry does not use SO2. Yes, they have hops, which are antimicrobial, but they're also really careful. And they sterile filter their products often. So the big breweries will pasteurize. But there's other options for antimicrobials. DMDC, Velcorin is another option. The problem with completely eliminating SO2 is its antioxidant capabilities. We do not have anything yet that can completely replace SO2's role as antioxidant. There's been work trying to replace it with things like glutathione. There is nothing that can serve as an effective antioxidant and a scavenger, especially for hydrogen peroxide, as SO2. And people who've tried to replace it with other things like vitamin C, ascorbic acid, glutathione, you do not prevent formation of oxidized aromas. So for right now, we're still using SO2. Okay, bound and total. We have additional fractions to be concerned about. So we have molecular SO2, bisulfite. We talked about that. It's free SO2. Some portion of this bisulfite will bind, it'll form chemical bonds to other components in wine. This is referred to as bound SO2. And then the sum of all of these forms together is referred to as total SO2. So bound SO2, it doesn't help you very much with antimicrobial or antioxidant activity, where it may provide some advantage, actually, is that it can serve as a reservoir. It's a backup source of free SO2 during storage for some forms of bound SO2. But generally, people consider bound SO2 to be a problem. It counts against you for TTB regulations, but it doesn't help you as an antimicrobial or an antioxidant. So here's some examples of binders, and they vary in how strong and how weak they are. So glucose in sweet wines can bind SO2. Anthocyanins in red wines, that's pigments, can bind SO2 and they'll get bleached. Methionine, acetaldehyde, some oxidized smelling compounds can bind SO2 and become odorless. So there's a broad range of species differing in how strongly they bind. So glucose, most of the sugar in a sweet wine is not bound up to SO2, but almost all of your acetaldehyde would bound up in a wine that's got SO2. So the reason to measure total SO2, well, number one is you have to, you're required to, it's regulated. You need to make sure you're under the legal limit, which is a regulatory limit. Bound SO2 can inhibit malolactic fermentation. So if you had a lot of SO2 prior to fermentation, in particular, for example, SO2 added to the grapes, you'll bind it up. This can inhibit subsequent malolactic fermentation. But one of the main reasons to measure this regularly, every time you measure free SO2, also measure total SO2, you can use it to determine the soundness of your containers. So as an example, this is data from a bag-in-box study. And if you look at free SO2 versus total SO2, over time, this is a wine that's getting slowly oxidized. We have this initial drop in SO2 coming about from oxygen in the headspace. SO2 decreases linearly over time. You can use this decrease to calculate how much oxygen is getting into the wine over time, how much oxygen per day. If you look at free SO2, it's changing over time. And that's because some of the total SO2, this bound SO2, will, as free SO2 is consumed, it'll release bound SO2 and reform free SO2. Looking at changes of free SO2 is not a valid way to estimate oxygen ingress in your wines. Looking at total SO2, the rate of total SO2 loss, is a valid way to quantify how much oxygen is getting into your wine during storage. So measure total SO2 also. This is a wonderful diagnostic tool for figuring out if your containers, whether it's a tank, or barrel, or a package like a wine bottle, this is a way to evaluate their soundness and how much air they're letting in. Okay, and finally, I can skip this. I'm going to move forward. This will be in the final slide, sorry, the slides that get distributed. I think that they'll be available if you'd like to learn more about what typical oxygen ingress rates are for different packages. So some last final sort of disconcerting news. We talk about free SO2. Katie will be discussing free SO2 in greater detail in her talk following mine. But free SO2, at least the way that it's usually measured, does not always mean free. So here's typical ways that free SO2 gets measured. Katie will be talking about all of these more, some of these more, talking about things like ionometric titration, or Ripper titration, maybe fusion type titrations, aeration oxidation, or AO. All of these standard methods, the sample is first acidified and usually also diluted. When you acidify and dilute a wine sample, as it's done for almost all of the traditional methods, you will break up some of the bound SO2 complexes to form more free SO2. You will measure more free SO2, whoops, it's all there, more free SO2 than is actually there. So during a typical analysis, you start off with bound anthocyanins, you do your analysis, you decrease the pH, you dilute the sample, you end up releasing these anthocyanins. And more importantly, you free up your SO2 that was actually bound. So in all classic methods, we are overestimating how much free SO2 we actually have. So what we've been working on in my lab for a few years now, is how do we measure free SO2, either bisulfite or molecular SO2, without having to change the pH of the sample? And what we've been working on is measuring it in the gas phase, where you can smell molecular SO2. A small portion of it is gaseous. So we're measuring what your nose measures. And the way we do that is with these gas detection tubes that are produced for the mining industry and for environmental monitoring. They also are called, they're made by a number, they're called Draeger tubes or gas tech tubes, named after their manufacturers. And the idea is you take a plunger, take a plastic syringe filled with partially with wine, equilibrate, and then expel the headspace through the gas detection tube. And there's no perturbation, no change in sample pH, no change in volume due to dilution. You're measuring only the true SO2. And when we compare these values between what we get from a standard method, like AO, aeration oxidation, and headspace GDT, or our non-perturbing method, what we see is that the amount of free SO2 or molecular SO2 that's in a red wine is overestimated by typically a factor of 3 to 4 in red wines. Put another way, most red wines have negligible amounts of free and molecular SO2. So what we're calling free SO2 in most red wines is really better thought of as apparent free and molecular SO2. With whites, rosés, because you don't have this anthocyanin binding, the numbers seem to line up pretty. Okay, so does it matter? What we think right now is that if what you care about most is long-term oxidative stability, will my wine oxidize or not? Then measuring this SO2, this apparent SO2 with the classic methods is entirely appropriate and probably more appropriate because the anthocyanin-bound SO2 can get released during storage as free SO2 is consumed. This anthocyanin-bound SO2 acts like a reservoir, a backup source of free SO2. So for most wines, this is your number one concern, is making sure that you've got enough SO2 present to prevent oxidation. So it makes sense to keep using the standard methods. There's a few times that we think that headspace GDT might be a better metric. Number one is it does seem to predict microbial stability a little bit better. It definitely predicts the sensory effects. You will not find a red wine that singes your nostrils unless you added impossibly high levels of SO2, illegal levels of SO2. Typical red wine, even if it says that it's got, I don't know, 2 ppm 2 ppm molecular SO2, that should be enough to singe your nostrils. You won't smell it in a red wine because it's not actually free, not actually. And finally, we think this is a better way to predict reactions involving SO2 and their rate, such as H2S in canned wine. SO2 can react with can, we think by going through the can liner, and the true SO2 is a better predictor of its reaction rate as opposed to the apparent SO2. This is also one of the reasons why it's pretty unusual to see, if you see a canned red wine, they usually don't have H2S, while it's quite common for ciders and for white wines. Okay.
So Gavin, I would say just 2 minutes remaining.
Oh, good, perfect.
Please.
Thank you. We were discussing what's the most kindest, gentlest way to tell some of the times on the stop. And that's probably the kindest, gentlest way of doing it. Thanks, Ed. Okay, so very quickly to wrap things up. From our research, first of all, the commonly used methods all badly overestimate free and molecular SO2 in red wines because they're also measuring the anthocyanins that are bound to SO2, or say the SO2 is bound to anthocyanins. This anthocyanin-bound SO2 does not seem to have much of any antimicrobial activity. So this may not be a problem because the standard methods do a perfectly good job for predicting the antioxidant capacity of SO2 still. And you don't want to start dumping in large amounts of SO2 into your red wines. You will no longer have a red wine, you'll have an orangish wine because the SO2 will bleach your color. What I recommend instead is, this may help explain why even when you've got a high pH wine, you try to get your SO2 adjusted just right, you can still have problems with things like Brettanomyces because you don't actually have any SO2 in those wines. So take extra special care with your darkly colored red wines. They are probably going to have very low levels of truly free and truly molecular SO2, perhaps membrane filtration, perhaps DMDC, perhaps more frequent checks in the winery. But keep in mind that your SO2 for those darkly colored reds is probably much lower than you think it is. Okay, so wrap up my final minute. 2 separate constraints: free for preventing oxidation, molecular for preventing microbial spoilage. There's also total SO2 for measuring oxygen ingress and monitoring the soundness of storage containers. And I think this is a good time to wrap up and hand off things to Katie so we can stay on schedule.
Thank you very much for your great presentation. We have a good overview of sulfur dioxide management, and now we will have more information about what to do in the winery with Katie. So I'm leaving the room to introduce Katie Cook.
And that is my pleasure. Thank you, Dr. Sacks, for that fascinating research information. I can't wait to hear more about headspace gas measurement in the winery. Katie Cook is now going to talk about practical winemaking aspects of sulfur dioxide. Katie is from Scott Laboratories, has a wealth of winemaking education and experience, particularly in cold hardy varietals. Katie has a BS in biology from the University of Texas, Houston, and a master's degree in enology from the University of Burgundy, or Bourgogne, in France. Prior to joining Scott Labs, she was the enology project leader right here at the University of Minnesota. And most recently, she's also the assistant winemaker at Herman J. Weimer Vineyards in Dundee, not Dundee, Oregon, Dundee, New York. When Katie's not taking time talking with her clients, she enjoys gardening and spending time with her winemaker husband and young daughter. I just want to also say personally that Katie was one of my very first mentors in cold climate winemaking when I moved from California to Minnesota 10 years ago. She continues to be an excellent and reliable source of information and truly a treasured friend and mentor. You may go ahead, Katie.
Thank you for that. I think
So you can start sharing your screen if you want.
yeah, I thought I did, and I think I shared the wrong thing. Sorry, I think.
Yep.
Was I sharing my my email?
Your mailbox, yeah.
Okay, now I'm sharing PowerPoint. Okay, well, thank you for that introduction, Drew. That was very kind. And thank you for Gavin for starting off with your good introduction to sulfur. I mean, I was told more to approach really a practical aspect of SO2. So I'm going to try and dive right into this more from a winemaking standpoint and using sulfur dioxide in the winery. So not as intense chemistry as Gavin, but that stuff is really important to know. And I always learn something listening to Gavin, so it's great. Okay, so just a reminder, the two main functions of SO2 is antioxidant and antimicrobial in your winemaking. And I say this for anything that you add into wine is always think about your reasoning for adding it. So sometimes you're adding your sulfur for the antimicrobial protection, and sometimes the most important reason you're adding it is for the antioxidant protection. And so always think about that when you're adding your addition, and think about that with any wine addition, because that's more conscientious winemaking. The times to consider adding your SO2. So these are more of your critical moments where you're going to be adding it. So at harvest, maybe in your picking bins would be one time to think about it. During pressing of your white grapes or crushing of your red grapes. Post-alcoholic fermentation, post-malolactic conversion, during your aging and storage, and then prior to bottling. These are all times to, check, adjust levels of your sulfur. Most of the time when wineries are adding sulfur, they're adding it as a salt of potassium metabisulfite. Remember that your metabisulfite, since it's a salt, chemically, it's only about 57% sulfur by weight. So when you're calculating your addition, it's a very easy calculation. You take your desired concentration of sulfur, you divide it by that 57.6%. You multiply it by the volume of your wine in liters. If you're doing gallons, you have to do another conversion step. So just go with the metric system, and then you'll get the amount of, or the mass of potassium metabisulfite to add. A lot of winemakers might even round that down to 50% to make that math even easier. And another reason people might do that is keep in mind when you buy that bulk KMBS, over time it can lose efficacy, especially if you're opening and closing it in a wet environment. If that salt gets wet, that sulfur can get used up a little bit. So you might not actually have 50% sulfur by weight. So it's always a good idea. Buy a new container of KMBS at the beginning of harvest every year. You don't have to throw out your old one. You can use that for cleaning or for flushing your filter sheets or whatever. There's a lot of uses for it, but just make sure that you're using fresh KMBS all the time. So there's a nice quick calculation, an example of doing that. So if you want to add 50 milligrams per liter, which often we'll say parts per million, they're interchangeable in wine terms, and you have 500 liters of wine, the calculations at the bottom. So 50 divided by 57% times 500, 43,000 milligrams or 43 grams of KMBS would be how much you'd want to add to your wine. Usually you'd mix that, then you'd dissolve it in a little bit of water, add it to your wine, mix your tank really well to get it well distributed within your wine. Okay, so now going to the critical moments of adding it. So adding at harvest, this isn't always done. Sometimes when you might want to think about this, so especially machine harvested fruit in hot weather would be a time when you're, sorry, of course there's an alarm that goes off right when I'm giving my presentation. I live next to a fire department. So, the machine harvested.
That's okay, we can hear you.
Okay, don't worry, there's no tornado coming. I know you're in Iowa, but that's it. The machine harvested fruit. So when I was working in Australia, that was really common machine harvested fruit in hot weather, that would be transported long distances. So they tended to add quite high amounts of sulfur in the picking bins, where we then had to remove it later on, and I can talk a little bit about that as we go. But another reason you might want to add it in the vineyard is if you have some disease pressure in the vineyard. So that top picture, that's either insect damage or bird damage that's then compromised the fruit, or you might have sour rot, so you can have some acetobacter issues. And like Gavin said, the nice thing about sulfur is that bacteria is pretty sensitive to it. So you don't need a lot of SO2 to kind of knock back that bacteria population a little bit to get it under control. And then another thing is, if you know, like, so if you're harvesting your fruit, a lot of people will pick in those small picking bins, but more commonly is what you see here in this picture is where it might be hand harvested, but then dumped into a larger macro bin. And the weight of the fruit starts crushing the fruit on the bottom. So you start getting juice on the bottom that can be exposed to bacteria or native yeast or spoilage yeast. And if you aren't able to process that fruit quickly, if it's on a transport truck or sitting on your press pad for a couple hours, that can start to spoil pretty quickly. So those are times when you might think about doing it at this point. But again, it's a decision-making process that you can do. The second critical point would be at crush. So this is or at pressing. So this is for white and rosé wines. You're going to add it directly to the press pan at this point. So you'd mix up a solution of sulfur. And usually you would calculate, a good way to do it is to calculate potential yield. So for white wines and rosé, you're typically thinking about 600 liters per ton. And you would base your addition based on that. This is one of the main reasons for doing this is because this is really a period of high oxygen exposure in your wine. So you're looking at that antioxidant potential. That being said, there's a lot of winemakers that are looking at hyperoxidation of the juice, and it's a stylistic choice. You can look into different reasonings behind that. I won't go into it, but generally, if you want fresh fruity wines, you are adding SO2 at this point to help remove some of those byproducts of oxidation. And another reason to add it at this point would be when your fruit quality is suspect. So if there's botrytis, botrytis can have an enzyme called lactase that has some oxidative capabilities on the wine. So it can help knock back some of that. Sour rot is another important one. So if you suspect that there's a high bacterial load. This SO2, so you want to be a little bit careful because you don't want to add too much sulfur at this juice point. One of the reasons why you don't want to add too much is because it can interfere with the implementation of your yeast. So if your free sulfur is above 30-40 parts per million, you might have a hard time, your yeast might have a hard time starting, and then they'll struggle they might die right away, they might struggle starting, or they might just struggle throughout fermentation, and you'll have some reduction issues with fermentation. So be a little bit cautious with how much you're adding at this point. You can add some complementary antimicrobial treatments to your wine to help with that, like lysozyme or some chitosan products can help remove some of that bacteria as well. So those are other things to think about. Oh, and I had kind of a target rate. So 20 to 80, if you're going at that higher end, that 80 parts per million dosage rate, that's more if you have really compromised fruit that you're worried about. In general, you would stay on the lowest end as possible with your additions at this point. Another thing to be aware of, I know Gavin's lab has done a lot of work on total SO2 at the start of fermentation, how it can affect some reduction aromas in the bottle or later on in your winemaking. And I think that was starting at about 70 parts total. So also be aware of residual sulfur coming in from the vineyard and adds that happened during harvest. With red grapes, it's a little bit different. The the antioxidant protection of sulfur is a little bit less important at this point because red grapes have a natural antioxidant protection from tannins. And also you're processing the fruit a lot faster. So typically you destem, crush the fruit, and you're pitching yeast right away on this. So there's not the 24, 48 hours of settling where you're worried about spoilage. As long as it's coming in and being processed quickly, you probably don't have a need for sulfur on your red fruit. Where you want to think about adding sulfur is when there's disease pressure that's high. So again, the botrytis because of lactase enzyme. So it can help inhibit some of that lactase enzyme, or if you think there's some bacteria on the fruit. So you want to protect it from spoilage organisms. Another reason to protect from spoilage organisms, if you know that you're not going to be able to pitch the yeast right away. So if you're doing a cold soak on the fruit, or if you know you're not going to process it immediately, mostly the cold soak is going to be what you're worried about on this. And then also you want to be really cautious about your sulfur interfering with your malolactic fermentation. So really anything if your total sulfur is above 50 parts per million, that can interfere with your ML later on. And usually with red wine, 99% of red wines were worried about it getting through ML. So in general, when just differences between approaching your sulfur adds in white and rosé production, with the white and rosé wines, that antioxidant protection is a little bit more important because of the tannins in red wine having some antioxidant protection. You generally are more worried about the antimicrobial effect. And this is something to be aware of with hybrid grapes that are low in tannin as well, and even some vinifera that's low in tannin and Pinot Noir. You might need to use a little bit more than for high tannin reds. And then just remember that red wine generally undergoes malolactic conversion, so you want to be careful about adding too much sulfur early in malolactic fermentation, or early in the processing to avoid that. Sorry. Okay, the next critical point, so your wine is finished fermentation and you're trying to decide how much sulfur to add at the end of fermentation, or adding your sulfur at the end of fermentation. This is one of the most critical points. Generally, your wine is supersaturated with carbon dioxide, so you're not going to be so concerned with the antioxidant protection right away at the end of fermentation. There's also a lot of dead yeast in the wine that can bind SO2. So don't get so excited that you see your hydrometer, your Brix is at 0, and you're like, all right, my wine's at 0, I'm going to add my sulfur. You want to really make sure that your wine is at the sugar level that you're hoping for. If you're hoping for a dry wine, get an accurate measurement of that residual sugar. So even if your Brix drops below 0, you can still easily have 20-25 grams of residual sugar in your wine, which is going to be very hard to get rid of if you've added sulfur to it at that point. And same thing if it's a wine that you put through malolactic. Make sure you have a good lab analysis of your organic acids, of your malic acid residual, just because the chromatography method isn't very accurate with that. Again, if I get a phone call from someone that's saying, oh, I need to restart my ML, it didn't quite finish. The first question is, how much sulfur do you have in there? And if you have more than 10 parts of free sulfur and more than 50 to 60 parts of total sulfur, you're not going to have a chance getting ML to start again, just because the bacteria is very sensitive. I have this chart on here showing just the legal limits of sulfur for different countries. Unfortunately, and this is for total sulfur adds, and the US unfortunately doesn't break it down into wine types. We just have 350 milligrams per liter. I like to look at the European Union regulations for total sulfur limits, because that's kind of a good guideline for how much total sulfur you should be adding to your wine over the course of its life. So if you have a dry red wine, so dry less than 5 grams per liter of sugar, and it's gone through ML, there's not much risk for microbial spoilage in this. So you won't need very much sulfur over the life of this wine. So at the end of fermentation, at the end of malolactic fermentation, you might be adding 50 parts, anywhere from 50 to 100 parts, depending on how much you added up front on your wine. If you have a wine that's really sweet, say 6% residual sugar, you likely are going to be in the upper legal limits of your total sulfur addition. And a lot of that initial sulfur that you're adding at the end of fermentation is going to end up in that bound form of sulfur. So it's not uncommon to add 100 to 200 parts right away at the end of fermentation to make sure that you're not starting again. And that's common winemaking practice. And the more you have experience with this, you're going to get a better feel for how much to add at the end of fermentation. Here's another chart that Gavin talked a little bit about, how you have how at a certain pH you only have a certain percent of your free sulfur that's acting as molecular sulfur. So this is the percent of your free sulfur that's actually protecting against microbes. As he said, you're going to have different recommendations all the time. In general, we say 0.8 parts of molecular SO2 is what you need to prevent the wine from fermenting, from re-fermenting, or from having microbial issues. And so a chart like this is something you should print out and have on your wine, like pin to a wall in your winery just for a reference. And the biggest mistake I see people make is they'll look at this chart, which is great, to try and determine their sulfur adds. And they'll say, well, my pH is 3.4, my target free sulfur is 35 parts per million. And so I'm going to add 35 parts of sulfur. So they do their calculation and they add their 35 milligrams per liter of KMBS. And then they measure it afterwards and they have 10 parts of free sulfur. So that initial addition that you add, a lot of it is going to end up in that bound form of sulfur instead of this free form. So it's actually a better idea to overshoot this on that first addition to get as much of that sulfur in that bound form as possible. And then your subsequent additions will be smaller additions. And it sounds counterintuitive, but if you keep adding smaller additions, your overall sulfur adds are going to be higher than if you just started out with a big addition up front. So let's say you targeted a little bit higher on here. So you said, well, I want to have 35. Oops. So I'm going to add 90 parts of free sulfur. A lot of that'll get bound up. Maybe you end up with 70 parts of free sulfur, which, as Gavin pointed out, the threshold for free sulfur is around 2 parts molecular. So if you're at 70 at 3.4, you're almost at that threshold. So that wine is going to smell strong of sulfur at that 70 parts. You're going to think that you made a big mistake. You're going to be worried about it. But also remember that every time you move that wine, you're going to lose anywhere from 5 to 10 parts of free sulfur, and how Gavin had mentioned that earlier too. So that first addition is probably not going to be the last time that you move your wine during that racking. So you're going to be filtering your wine, you're going to be doing subsequent rackings, and every time you move the wine, you're going to be dropping 5 to 10 parts. So if you're going to filter 3 times, you're probably going to drop it down closer to that 35 parts. And this is the part that you need to have closer to bottling as well. So that first addition, if it's a little bit high, it's usually not super critical unless you go way, way over the top, which as long as you're following this chart, you should be okay with that.
Katie, you will have only 2 minutes remaining just because it won't be enough time for questions. Sorry.
Okay. All right, so I should be able to finish up. So the next critical point is in storage and aging. And So just during storage, it's really going to depend on the storage vessel that it's in, but really you need to remember to measure and adjust regularly. So with oak barrels, pretty much every time the barrel is topped, which is every 2 to 3 weeks, you're going to adjust sulfur levels. Some people think that's a lot, but there's a lot of oxygen exposure and headspace in barrels that happen. Plastic tanks, they tend to have a little bit more oxygen ingress, so you're going to be measuring and adjusting monthly or anytime your wine is moved. Stainless steel quarterly or anytime the wine is moved. And really at this point, this antimicrobial effect, is very important. The SO2 isn't, I think Gavin touched on this, it's not really a good antioxidant. There's other things you can add to your wines that work well as antioxidants. We do have, well, there are some yeast derivatives that do a nice job with it, that you can work on, or tannins or anything else. But really you want to worry about sparging, keeping your tanks topped, and keeping your exposure to oxygen at a minimum to protect against SO2. Okay, so measuring, I just will say this, there's basically 2 methods. There's an additive method and a separative method. I won't go into detail, but the additive method is, since I'm running low on time, is essentially a ripper. The separative method is aeration oxidation. And these are 2 methods that you should really learn for a small winery. The ripper method, it's essentially a titration, quick and easy to learn. If you took high school chemistry, you should be able to do ripper method. If you didn't, you can find some kids that look like about their age, and they can teach you how to do some titration. So that's really the positives of it. You'll see other Titrettes, Hanna Instruments, Vinmetrica, they're all based on this ripper method. So it's very common in the wine industry to use this. The downsides with it is, since you're looking for a color change in the wine, it's really hard to see that color change in red wine. So it's pretty inaccurate in red wines. You have other compounds that interfere with it, and you generally have a margin of error about 10%. So it's a pretty unreliable method. But I will say that it doesn't mean that you shouldn't have this in your winery. So this, because it's so quick and easy, it's a really good way to get a quick, dirty idea of what your free sulfur is in your winery, or what your sulfur levels are. The aeration oxidation is going to be the reference method. That's what the apparatus looks like for it. You separate the SO2 gas, it goes up this tube, collects with hydrogen peroxide here, and then you measure it. It's a little bit more complicated for learning how to do it. There's some training with it. The test itself is time consuming, but this is going to be the most accurate for measuring in the winery. This is another method, a newer method that Gavin talked about, using his headspace gas detection tubes. I will say that with all of these measuring methods, there is some discussion right now in the wine industry of what are best methods for measuring in the winery. In large labs, they're looking to newer methods with more expensive equipment that may be more accurate, given some of the reasons that Gavin outlined with some of the interference that can happen with some of these methods. And the final method that I will just touch on briefly, just because a lot of wineries are investing in spectrophotometers these days, is that you can do an enzymatic reaction and measure your sulfur by calorimetry. It does take some training to be able to do it, but if you have a spectrophotometer in your winery, there's a lot more that you can do with it. You can do your organic acids, your alcohol, your residual sugar. And so it's a nice tool to have to use in the winery. All right, so in summary, it's just that these accurate measuring, in addition, is its important skill for winemaking, and you should really be aware of the critical times when sulfur addition needs to be considered in your wine. All right, thank you. Hopefully I finished in time for questions.
Thank you very much.
Yeah. So, and I'm sorry, Gavin, you kept saying I was going to touch more on free and total, and
Oh,
I I
Pardon me.
wasn't.
Yeah, I was guessing wildly. So I was like 95% correct about what you would talk about. So let's focus on that part and not the 5% I was wrong about. So the next time, actually, while I'm saying those things, it'd be better if you just like quickly dash together some slides to add to your talk, just so I don't look foolish.
All right. Well, thank you, Katie. That was awesome. For the participants and viewers, now is the time to type your questions in the Q&A box or in the chat box for either Dr. Gavin Sacks or for Katie. We've got, about 5 or 10, 15 minutes to answer your questions. Before switching on to the questions, I have to state that some of this presentation of this material is based upon work that's supported by the National Institute of Food and Agriculture, U.S. Department of Agriculture, under an award number through the North Central Region SARE, or Sustainable Agricultural Research and Education Program, under project number ONC 19-053. The USDA is an equal opportunity employer and service provider, and any opinions, findings, conclusions, or recommendations expressed in this publication are those of the authors and do not necessarily reflect the view of the U.S. Department of Agriculture. Thank you for letting me get through that. We can start with your questions.
Okay, I was checking how I can unmute myself. Thank you very much, Gavin and Katie, for your great presentation. So we have quite a few questions so far. So let me check in the chat box. Okay, so the first one is, some of us are hobbyists, making wine on a very small scale, like 6 gallons at a time. So we need to do chems on a small scale too. If I want to apply some to grapes at harvest or crush, is it best to use a handheld spray bottle? And if so, how do I know how thoroughly to spray the fruit? And how long will it allow me to hold the fruit before crushing?
I would say that you can do a like a 10% or a 1% solution of your potassium metabisulfite and do your calculations that way. And that's how a lot of hobbyists will end up figuring out how much sulfur to add to their wine. So you just do a really dilute solution of metabisulfite. And you can find calculations online to figure out how to make a 1% solution for that.
And at the scale
like
that you're that you're describing, it's not obvious that you need to spray. You should be able to mix it in, homogenize it pretty easily. So as opposed to like if it's going to a large bin where you might not have access to the entire bin. So yeah, I agree with Katie. Spray bottle seems like it's not going to be a very accurate way to deliver what you need.
Because I think we have another question related to this one. Oh no, that was the same, sorry. So someone was just asking about the preferred method to measure SO2. I think, Katie, you started to talk about this preferred method to measure SO2.
So I don't know, what's the question, to explain more about Ripper method?
It's more like, what would be the favorite method to measure SO2 in a wine?
So it depends on how accurate you want to be.
And how much money maybe you want to.
And how much money you want to spend.
Do I understand? So tell me, so for Iowa, Minnesota, are most wineries doing analyses themselves, or do they have options for external labs they can send to?
Okay, so in Iowa, there is the Midwest Grape and Wine Institute, where they have fee-for-service for SO2 measurement, and I know that some wineries just request these measurements to the institute.
And I can speak for Minnesota. We run the gamut. Some small wineries are just still using Titrettes. Some are starting to use the Vinmetrica. When they do use the Vinmetrica, I try to remind people test it twice to confirm your findings. Make sure your reagents are fresh, aren't expired in their date. And the other thing that's really handy for the Vinmetrica is to get one of the expanded 10 milliliter burettes that's marked in half milliliter sections. And so you can increase your accuracy a bit by using that expanded scale burette. Certainly aeration oxidation is way more accurate, but as Katie noted, the equipment's a bit more expensive, and it does take a while. It can take you 20 minutes to an hour to get one result. So at that point, it's almost best just to send it off to a lab.
Yeah,
Like
I mean, for me personally, working in a winery, I like to have a combination of different techniques. I think the Ripper method is really nice for your everyday, just like I want to get an idea of what my free sulfur is. And you know that it's not accurate. And as long as you know that you're within that margin of error, it's more when you need to be accurate. So as you get closer to bottling, that's when you would want to use aeration oxidation a little bit more. But Ripper is a really nice, easy method. A lot of people, there's a lot of strong feelings between winemakers on like what on whether they like Ripper or not. I like the titration with Ripper just because I'm confident in my titration. I am not confident in Hannah and Vinmetrica just because I think there's a calibration that where things can go wrong, and how Drew said, your reagents, can be issues with the reagents. But I think it's a very personal thing with winemaking, but definitely as you get closer to bottling, you need to be as accurate as possible.
So I will add something to all this. Yeah, Ripper most certainly inspires strong feelings. So we had done a survey, done a review of some work that's done on round robin studies along with Sue Ebler at UC Davis. So these are same wines get sent out to multiple wineries. Everyone sends back their results, and ideally everyone's getting the same numbers, regardless of method. There's a tiny bias. There is a bias. There's a tiny bias between Ripper and AO and related methods. However, the accuracy of AO, and I should say the precision of AO, the variability, is as bad with AO as it is with Ripper. This is not because AO is a worse technique or a better technique. It can be better, but in practice, it's not, because AO has a high, I think the technical term is screw up potential. So a badly done AO versus a well done Ripper, I'll take the well done Ripper. My recommendation to winemakers is, so you're making numbers. How do you know they're any good? If you don't know they're any good, just send them out to an external lab. And the way you know if your numbers are any good and that you're competent as an analyst, take a commercial wine. First, you can participate in a round robin testing. The easier is take some commercial wines, two, maybe, send them off to Iowa State, send them to ETS, whatever's convenient for you. Then measure them in-house. Compare your numbers. If you do not know if your numbers are accurate, then why are you routinely relying on them? It does not matter if you use AO or Ripper if you don't go to the trouble of validating your accuracy with real wines. So that's my strong opinion. If you're not going to determine your own accuracy, then you should just send everything out externally.
I agree with that completely. And I worked in a Gallo cellar for 3 years, and they did that every quarter, where they sent out a commercial wine to all of the various Gallo facilities. And it was almost a competition to see who could get as close as possible and could repeat themselves every time.
Yeah, hey Drew, you know that our students at Cornell actually participate in that robin testing too with Gallo.
Oh, that's right.
To actually send those their wines.
I didn't know that. I was just
yeah. They don't do as well as Gallo.
I was just a lowly cellar tech for Gallo for a number of years, and I knew that the enologist, whenever that would happen, he was sitting on edge waiting for the results to see how he came out. Aude, are there any other questions, or shall I ask one or two?
There is one in the chat that if you want to ask, feel free to. Do you have access to the chat box, or do you want me to see it?
I have the chat box, but I don't see the question. Why don't you go ahead and ask that?
Okay, so there is one regarding a market wine. So I had a market that started at pH 3.36, and after fermentation it went to 3.99 and measured 6 ppm free SO2 based on chart. that meant a large addition of potassium metabisulfite, and it felt like too much. Can you talk about hybrid reds and their higher pH?
Okay, there seems to be two questions there, just to make sure I understand. One is you had this phenomenon of the wine starting at pH 3.3 and it went to 3.99. And then the second was, I missed the part about the SO2. It was a lot of SO2 was added, but now there's no free SO2. Is the comment?
So yeah, it was just to have more information about the addition of SO2 with the pH that is pretty high, like 3.99 after fermentation. And so this person measured the free SO2, it was 6 ppm. So I think they want to add SO2, but
It's a high
it seems to
pH.
be too much based on
So
the
how do you
pH.
treat SO2 with high pH wines?
Mm-hmm.
Yeah. Do we need this wine to stay at high pH, or are we allowed to remedy that? That's my first thought: is do we have to keep it 3.99? Or because if we can, I would love to. I'm not sure, Katie. I mean, you do more practical winemaking. I do the impractical stuff. But if it's possible, I'd like to see what the TA looks like too. But it's possible to add some tartaric acid and get some precipitation of potassium. Then that'll bring the pH down to something a little more reasonable. But if it's a very high acid Marquette, I understand there's some limitations sometimes with how much you can do with acid additions. But hopefully, the tartaric, you can get most of it back out in the form of precipitation and bring that pH down.
Mm-hmm. Because I have another question from maybe the same person saying that is that typical for a hybrid red to go that high in pH?
So I saw that pretty regularly in Minnesota. I think I think I was shocked the first time I saw something like that where it went from, I don't think I saw that big of a jump from 3.3 to 3.9, but 3.5 to 4.2, I guess that's kind of the same, but going up pretty quickly. And I would always just adjust, I would acidify early on, just so that you stayed below a 3.6, just to make sure that you didn't go, but yeah, if you're against acidifying the wine, or if it's going to be too much acid, I would almost worry about just the antioxidant protection of the SO2 at that point, and not worry about having those really high free sulfur levels, and then worry about just having really good sanitary practices and getting the wine in bottle really quickly, and sterile filtering and getting in bottle. That's in Washington State, they deal with pretty high pH wines, and that's how they deal with it. So good sanitary practices.
I would recommend, if you're having problems predicting the pH of your final wine, when you have your original grape sample, instead of taking a nice gentle free run press, try to really macerate those skins, because the pH of the skins is higher than the pH of your free run, because there's higher potassium content specifically. So when you're prepping a red juice sample for analysis, you want to vigorously, treat it like a red winemaking, vigorously macerate that fruit. So that you can see what the final pH is going to be like, and that might help you with thinking through initial tartaric additions.
Mm-hmm.
And I will say, when I used to add tartaric, it was almost like a sacrificial tartaric addition, because usually with the hybrids, why they have that big jump is because there's so much potassium in them. And so I would add 2 grams of tartaric acid up front, and my final total acidity measurement wasn't any different. Because all that tartaric just drops out, it just pulls out your potassium without raising your pH. So I would just use, that was kind of my thing, I would use an acid addition as a sacrificial acid addition to lower my potassium.
Mm-hmm. And Katie, would that be assuming that even with a Marquette, as you once taught me, that you, through cold stabilization of that red wine, you can get some of that excess tartaric acid to precipitate out?
Yeah, and that would be the sacrifice. So usually, if it doesn't come out during fermentation, it drops out during cold stabilization.
Perfect.
And that 3.6 target that Katie's talking about is a good one to keep in mind. Number one, that's when above that was when pediococcus, spoilage organisms, become more of a problem. But also, that's the pH where potassium bitartrate is least soluble. So you're going to get the most out if you target that pH.
Yeah, it seems that we need another webinar about acidity.
You're in the
Absolutely.
Midwest. All of your webinars
Well,
should be
once
about
you learn
acidity.
chemistry with hybrid grapes, you can do anything with winemaking.
True.
I'm going to ask one more question that I get asked a lot.
Mhm.
Oh, I see. So Stan Stevens, yes, to answer your question, Stan, yes, it will. If you add to target, it precipitates out. Yes, because effectively you've just you've reverse titrated your wine. You've pulled up potassium bitartrate, so that'll bring down the pH.
So we are really out of time, but that's fine. We'll keep going because that's recorded, so we can always explain the response to the questions. So I have another question. The SO2 addition after fermentation, does it mess with the polymerization of tannin? Low tannin grape variety that you are adding tannin, like Marquette, should you wait to add SO2 till after tannin addition? So
Okay, so
maybe
when would this
you
be?
can precise, yeah.
Yeah, I got strong opinions about this one. So, so actually, oh, you must have strong opinions about this one, right? You do work on tannins.
Yeah, I do work on
What do you think
tannin.
about polymerization and tannins?
So I know that the SO2 can bind, so it's able to bind with the anthocyanins, as you explained, the bisulfite to form new compounds. And the SO2, we made, like, I was participating in a study where they were looking at the addition of SO2 to flavanols, to the monomers. It seemed to be able to interact as well to form new compounds. But then for the polymerization of tannin, I don't know really if that would mess up anything about the polymerization of tannin.
Yeah, and one of the problems is so the classic idea of what happens to tannins during aging is they like get bigger and bigger and bigger. And that's how they soften. The problem is that bigger tannins are more astringent. So clearly it's not happening. And the other is if you look at the average molecular weight of tannins during storage, it goes down, not up, because the tannins are getting big, long molecules to begin with, and they're getting chopped into smaller and smaller pieces. So once they do that, Aude, as you said, those tannin fragments will react with anthocyanins and make polymeric pigments. They'll react with SO2, and probably that contributes to some of the softening of tannin. This is an active area of research, is what I would say. It's not. This is, and so it's obvious that it's not that what's happening with tannins is not just polymerization. In fact, it's the opposite of it. It's depolymerization. What we don't know is how do all the other things in wine affect the final outcome. So SO2, anthocyanins, other tannins, other polyphenols. Almost certainly SO2 plays a role, but wow, for me to give you a specific recommendation right now. I mean, the one thing I would say is that if you're doing very high SO2, if you have higher SO2 and you're doing micro-oxygenation, I don't really understand what you're doing because the SO2 would scavenge all the products in the air. So if you're going to go if you do micro-ox, then it makes sense to have SO2 a little bit lower because I'm not sure what you think is going to change with the wine.
And for the tannin, we still don't really know, or we still don't understand how oxidation or aging lead to a different astringency perception, just because we don't know yet, like if the tannin are oxidized and they just precipitate because of the structure that change, or if it's just a reduction of the size, like a cleavage of the molecule of tannin. Yeah, so SO2 and tannin, I don't know.
Yeah, probably having red wine, really high SO2 to begin with isn't, as Katie said, there's a limit to what you want anyway, because eventually you'll turn your red wine into something else, into a orangish wine.
Okay, and I just had a question. What do you think about using an alternative to SO2 in combination with SO2? So not still using some SO2, but with another like chitosan, just to reduce the use of SO2. What do you think about that?
Yeah, I think that's a good methodology for reducing your level of SO2. And I mean, I can talk about products that we have at Scott Labs, but There's a new one that's called, and I don't want it to be a sales thing, but it's called Glutastar, and it's called Glutastar because it's high in glutathione, but that's not the coolest part of it. It actually scavenges some quinones. And in some of it, I like it because my alma mater, University of Burgundy, was someone who came up with it, who helped with the research involved with it. And it actually, if you compare it in a control against sulfur dioxide, it has a better protective effect against oxidation than sulfur alone. So that's a really good product to use. Tannins are also another, some commercial tannins are nice because they scavenge some oxygen as well. So there are ways to lower your sulfur use by using some of these complementary. But it depends on why you're using the sulfur too. Is it antimicrobial or the antioxidant as well?
Yeah, I think, I mean, that's the key. So with Katie's comment about chitosan, that's an example of when you're concerned about antimicrobial. So for high pH wines, you have plenty of free SO2 for free SO2 for oxidation, but you can't get to molecular high enough based on that 0.8 ppm recommendation. And so those complementary tools like chitosan, like sterile filtration, like Valcaran, other things make a lot of sense. Where I think there's still dispute. There's interest. There's a amount of research and strong opinions is how much realistically can you replace of the antioxidant function of SO2 over the long term? And I mean, SO2, as Katie pointed out, like it does not react directly with oxidation products, but it does eventually. And so this is the big long-term question: is you store a wine long enough, how much can you really replace of that SO2 if what you want to do is limit its need for preventing oxidation? It's almost certainly going to be more than, you need more than zero, but you may be able to limit it.
Okay. Thank you very much to all of you for attending, and to all of our presenters today, Gavin Sacks and Katie Cook, and Drew for co-hosting and co-organizing this webinar. So remember, we will have, you will receive a link to a survey to complete. Please take a few minutes to complete this survey. Also in the future, because we know that now harvest is starting, so everyone will be very busy with winemaking. So we will keep doing the research and winemaking webinars. So maybe on manual lactic fermentation, filtration, aging, acidity, because it seems to be very important. So most probably by the end of October, beginning of November. So stay tuned for that. And if you have any question, feel free to reach out to us, either myself or Drew, or directly to Gavin Sacks and Katie Cook. And so thank you very much for all the time. Yeah, and your great presentation. Thank you all.
We're good. Thanks.
Thank you. Bye.
Bye. Bye, Aude. Bye, Katie. Bye, Drew.
Bye-bye.